Hello everyone, and welcome to this audio podcast. This audio podcast in on looking at Oxford Nanopore sequencing technology involvement in the genomic domain and its impact in the biomedical science field. And this overview is works that has some fantastic works that have been that have been articulated by a by a science student, Heidi Stefan, in one of the academic institutions in the southwest UK, and in continuing on from the introduction, now looking at function of nanopore sequencing technology, and beginning with, the sort of principal elements. So the principle of nanopore sequencing relies on the passage of bases. So, for example, adenine, cytosine, thymine, guanine, and or uracil from DNA, RNA through a nanopore device known as a min ion, and which determines the signal produced by the bases adenine, cytosine, thymine, guanine, or uracil, with the use of continuous electrical current passing through. Flow cells in the min ion technology contain the proprietary sensor array, application specific integrated circuit, and hundreds of nanopores where DNA, RNA passes through, and can be monitored. Um, and as part of the sample preparation, Oxford Nanopore sequencing technology provides a sort of a variety of library preparation kits to suit whole genome research or targeted genomic investigation. And an amplification kit is provided to allow or prompt sort of uninterrupted sequencing of short to ultra long fragments of DNA, which eliminates the requirement of polymerase chain reaction or PCR amplification, allowing investigators to use this kit along with low amounts or quality of DNA. An example is the ligation or splicing. An example is the ligation or splicing sequencing kit, which has a preparation time of in around about 60 minutes. 60 minutes or an hour. So RNA library preparation kits are also offered, which can be adapted to suit the research to allow a high accuracy, long reads, or fast turnaround times. An example in around this is the cDNA PCR sequencing kit, which takes about 225 minutes, including PCR time, to output over 100 m reads. High molecular weight genomic DNA samples, which are about 50 kilobytes larger than that, which are 50 kilobytes or kb larger than larger need to be handled with caution, and adapted techniques of investigations involve no vertexing, and it is recommended to use pipettes with wide bore tips to prevent or preclude the sample from being sheared. Example: the preparation for extracting genomic DNA from human brain tissue is completed through a particular sort of kit, a particular sort of a manufacturing kit, a manufacturing DNA kit, where it can extract anything from 8 to 15 micrograms of DNA from 25 milligrams of brain tissue through three stages known as tissue lysing, tissue splicing or lysis, and DNA extraction and DNA shearing. Once collected, a library can be prepared. A library can be prepared to allow the nanopore sequencing technology to work. Um, and so, in around this, a little bit in around how it works. So, when the base progresses, or when the base progresses through a nanopore, it disturbs the electrical current also passing through, resulting in an altered signal, producing a sort of a squiggle. Each distinct signal corresponds with a base, and it is recorded by the min no by particular software by particular software found in all nanopore devices, where it is stored in where it's stored in files, producing a sequence graph. These files of data are then decoded through base calling algorithms to determine the sequence, and create BAM files to allow investigators or researchers to evaluate and analyze. So that draws this audio podcast to a close. This audio podcast that looked at in around the sort of principle, in around the function of nanopore sequencing technology. The subsequent audio podcasts in this series will look at some of the advantages and disadvantages of nanopore sequencing technology. This audio podcast is now drawn to a close as part of the Storm Mode Isomer. This audio podcast as part of the Mayhem and Twister. This audio podcast as part of the Miliplex, and this audio podcast as part of the Storm and a Storm Robotics production.